Today we are going to study about chromosomal theory of inheritance. Gregor George Mendel in the year 1865 studied the inheritance of well-defined character of pea plant, but for several reasons it was unrecognized till 19,000. After the three scientists, Hugo D. Virus, Concons, and Teschmark independently discovered Mendel's result of inheritance of character. After the discovery of microscope, they discovered the nucleus. In eukaryotic cells, worm-shaped structure formed during the cell division, and it is called as chromosome. In organism which possess two complete basic set of chromosomes are known as diploid. A chromosome consists of long, continuous coiled piece of DNA in which genes are arranged in linear manner. So genes are arranged in a straight line. So only it is called as what? Linear manner. Each gene has a definite position on the chromosome. So each gene is having a specific position in the chromosome, and this specific position is called as locus. And the specific position is called as locus. The place where it is attached is called as what? Locus. These genes are the hereditary units. The genes are hereditary units. Okay, na? So chromosomal theory of inheritance states that Mendelian factors have a specific locus on the chromosome, and they carry the information from one generation to the next generation. Salient features of the chromosome. Somatic cell of organisms are derived from the zygote by repeated cell division, that is by mitosis. So here the body cell is derived from the zygote by repeated cell division. They consist of two identical sets of chromosomes. So they consist of two identical sets of chromosomes. One set is received from the female and another one from the male parent. So these two chromosomes constitute the homologous pair. These two chromosomes forms the homologous pair. Chromosome retain their structure, uniqueness and individuality throughout the life cycle of an organism. Each chromosome carries specific factors are termed as genes. Okay, no? Each chromosome they will carry a specific determinants which is now called as genes. The behavior of the chromosomes during the gamete formation provide evidence to the fact that genes are factors. Linkage. The gene which determine the character of an individual are carried by the chromosomes. The gene for different character may be present either in the same chromosome or in different chromosomes. When the genes are present in different chromosomes, they assert independently according to Mendel's law of independent assortment. Biologists came across certain genetic characters that did not assort out independently in other organisms after Mendel's work. One such case was reported in sweet pea, that is Lathyrus odoris, by William Bateson and Reginald Punnett in the year 1906. They crossed purple flower long pollen grain with red flower and round pollen grain. They crossed it in the first generation. In the F1 generation, they got purple flower and long pollen. From this, they came to a conclusion that this purple flower and long pollen is a dominant character, and this red flower and pollen grain, round pollen grain is a recessive character. So they crossed the F1 generation. So they crossed the F1 generation with the recessive parent. So we have to get the result in the ratio 1 is to 1 is to 1 is to 1. But it is not got according to the Mendel law. According to the test cross, we have to get that the ratio should be 1 is to 1 is to 1 is to 1. But in this cross, the ratio is different. Okay, a greater number of F2 plants have purple flower and long pollen, and red flower and round pollen grain were observed. So they concluded that purple flower, long pollen grain and red flower, round pollen grain are found together in the same homologous pair of chromosomes. So they do not separate it out during division. So they did not assort out independently. So this tendency of staying together during their separation is called as linkage. It is called as what, ma? Linkage. Gene located together in the same chromosome will inherit together, and such gene is called as linked gene. When two genes are sufficiently far apart on a same chromosome, okay, when the two genes are sufficiently far apart on the same chromosome, then it is called as unlinked gene or synthetic gene or SNP. To be differentiated by the value of recombinations of frequency. If the recombination frequency value is more than 50%, the two genes shows unlinked. When the recombination frequency value is less than 50%, they show linked. Closely located gene shows stronger linkage, while genes widely located shows weak linkage. When two dominant allele or a recessive allele occur in the same homologous chromosome, they will inherit together in the same gamete, and such thing is called as coupling or Cis configuration. Here, if a gene, a dominant gene and a recessive gene is present in the same homologous chromosome, they tend to inherit apart. They will separate out in the gamete formation, and such thing is called as repulsion or transconfiguration. Kinds of linkage. T.H. Morgan found two types of linkage. They are complete linkage and incomplete linkage. Depending upon the absence or presence of the new combination of linkage, it is classified into two types. One is complete and another one is incomplete. Now we are going to see about the complete linkage. When two genes remain together, they will result in they will observe only the parental combination is observed. Only the parental combination is observed. So here, when two linked genes are seen very close together on the same chromosome, and they do not exhibit crossing over, and this type is called as complete linkage. Okay, ma? When two genes remain together, okay, ma? They will show the parental combination. Only the parental combination is observed. When two linked genes are very close together on the same chromosome, they do not exhibit crossing over. So this is called as complete linkage. It is rare but has been reported in male Drosophila. C.B. Bridges in the year 1919 discovered that crossing over is completely absent in some species of male Drosophila. Here, when two linked genes are far apart, separation is possible. As a result, parental and non-parental combinations are observed. So here they exhibit crossing over, and this type is called as incomplete linkage. This was observed in males and it was reported by Hutchinson. Once again, if two linked genes are far apart, separation is possible. As a result, parental and non-parental combinations are observed. They exhibit crossing over, and this type is called as incomplete linkage.